Hello students, hope you are doing well for the NEET exam. Welcome to my channel Vivid Botany. We are looking here a question about the Rubisco engine. We know Rubisco is very popular. It's been asked so many times in the competitive exams. See here the question. When you see the word Rubisco here, you see here, are you BISCO. R is caps, B is caps, C is caps. So, Rubisco stands for ribulose 1,5 bisphosphate carboxylase oxygenase. It means ribulose 1,5 bisphosphate it is a what do you call pentose ketose sugar. It is a pentose ketose sugar. Pentose means it has got 5 carbons. 1,5 means it has got 2 phosphates. One is at first carbon, another is at fifth carbon. Ketose, you know, aldose ketose. It is a sugar molecule. Then why it is added with carboxylase and oxygenase? Remember, these two indicate that it's an enzyme first of all. You know, enzymes are organic catalysts, they are biocatalysts, they speed up the rate of reaction by decreasing activation energy. And why it is given the dual name like carboxylase and oxygenase? Yes, it is a dual enzyme. Dual enzyme means it can fix carbon dioxide and oxygen. Yes, it can fix both. Then when does it fix? Does it fix both at same time? No. It fixes either CO2 or O2. Generally, this enzyme shows much affinity towards carbon dioxide. Affinity means it likes. It likes to fix carbon dioxide rather than oxygen. Then when does it fix oxygen? Yes, that is a good question. When oxygen content is more in the atmosphere, when CO2 content is less in the atmosphere, that is, the relative concentration of CO2 and O2 determines the binding of oxygen to Rubisco enzyme, right? So, generally in atmosphere, we know oxygen is 21 percentage and carbon dioxide is 0.03 percentage. I wrote it here. But when CO2 becomes more than, when CO2 becomes less than 0.03 and O2 becomes more than 21 percentage, then Rubisco has no option but to fix the oxygen. Yes. What happens when Rubisco fixes carbon dioxide? And what happens when Rubisco fixes oxygen? That is, let us see. When it fixes carbon dioxide, normal C3 cycle takes place. Normal C3 cycle means, I wrote it here, you see, this is RUBP plus carbon dioxide in the presence of the Rubisco, where Rubisco acts as carboxylase. There, two molecules of 3-phosphoglyceric acid is formed. Each phosphoglyceric acid is a 3-carbon organic acid. 3-phosphoglyceric acid means, it doesn't mean that it has 3 phosphates. It is having only 1 phosphate at third carbon. Phosphate means phosphoric acid. Orthophosphoric acid present at third carbon of the phosphate. Now, so it is called 3-phosphoglyceric acid. Two molecules of 3-phosphoglyceric acid are formed. Each phosphoglyceric acid, as it contains 3 carbons, 2, 3 or 6 carbons are present here, and RUBP 5 carbons plus 1C, that is carbon dioxide, to give rise to 6 carbons. So, when Rubisco acts as carboxylase, a 3-carbon compound is formed. So, the cycle is called C3 cycle, and the plants are called C3 plants, right? Then, what happens when Rubisco fixes oxygen? Does it really fix oxygen? Yes, it fixes oxygen. Then when does it fix oxygen? I told you that when oxygen is more than 21, CO2 is less than 0.03, then Rubisco fixes the oxygen. Yes, generally you see that. For example, a park is there, a garden is there. In that garden, many plants are there. We went to the garden. We sat there for some while. Especially in the afternoon, for example, after 2 o'clock in the midnight, 2 o'clock in the midday, 2 o'clock in the midday, like 1 o'clock, 2 o'clock, 3 o'clock, suddenly we get suffocated. That is, we don't get enough oxygen to breathe. The same problem arose at some place in US. Then scientists have conducted investigation on that incident. Why CO2 concentration is increasing in that area, especially during mid-noon, during midday. They understood that all the plants present in the park are performing respiration, especially photorespiration during the midday. They surprised for a while. Why they are fixing oxygen? Why they are not fixing carbon dioxide? Because we know when they fix oxygen, they liberate carbon dioxide. They understood that initially what happens when the day begins, normal C3 cycle takes place. Normal non-cyclic electron transport takes place. We know that in non-cyclic electron transport, water molecule is splitted. When water molecule is splitted, we know that Z scheme or Hill reaction, photolysis of water. So when this water molecule is broken down, then oxygen liberates into atmosphere. And usually oxygen concentration increases in the nearby areas of the plants that too surrounding the mesophyll areas. And as the day progresses, light intensity increases, Z scheme, non-cyclic electron transport takes place, water molecule breakage down will take place more and more quantity of oxygen liberates into atmosphere. This increased concentration of oxygen will compete with carbon dioxide that is going to bind with Rubisco. I have told you that Rubisco is, it can fix both CO2 and O2. So when O2 becomes more than that of CO2, than their normal values, the Rubisco will fix oxygen in such cases. When Rubisco fixes oxygen, what happens? Yes, that is our point of discussion today. You see here, Rubisco fixing here, RUBP fixing oxygen and giving rise to two molecules here, 3-phosphoglyceric acid, 2-phosphoglycolic acid. This is 2-phosphoglycolic acid, GLY COLIC. I write it here, glycolate. It is a 2-carbon organic acid, right? 2-carbon organic acid where phosphate is present on second carbon. And 3-phosphoglyceric acid, just now I have told you, it is a 3-carbon organic acid where 1 phosphate is present on third carbon, right? So a 3-carbon organic acid, a 2-carbon organic acid, both are formed during the photorespiration, where RUBP fixes oxygen instead of carbon dioxide as Rubisco acts as oxygenase. This phenomena is called as photorespiration. Why should we call it photo? Photo means sunlight. Respiration means we know breathing. That is taking in oxygen, liberating out carbon dioxide. The same phenomena takes place in plants also during sunlight, hence the name photorespiration. Discovered by Dicker and Tio. Do remember DICKER TIO, Dicker and Tio. So we also call it as glycolate pathway. Why should we call it as a glycolate pathway? Because in this process, glycolic acid is formed. We know that here, apart from phosphoglyceric acid, glycolate is formed, glycolic acid is formed. So we call it as glycolate pathway. We also call it as C2 cycle. Why? Because we know glycolic acid contains 2 carbons. So it's called C2 cycle and glycolate pathway. C2 cycle and glycolate pathway, photorespiration, right? So what? See, when Rubisco fixes oxygen, what happens to plant? Why it is lost on part of the plant? That is we have to understand here. See, when Rubisco fixes oxygen, it will liberate carbon dioxide. So, what happens if plant liberates carbon dioxide? Yes, there the twisting is there. When carbon dioxide liberated by plant, actually, what is the ultimate end product of photosynthesis? Making of glucose. Glucose is made up of what? Carbon. What is the source of carbon? Carbon dioxide. But what's happening in this phenomena? Plant is losing carbon dioxide. So, when plant is losing carbon dioxide, the input of glucose will decrease. The yielding of glucose will decrease. Whatever glucose it is going to prepare in the day, that amount of glucose will decrease by the plant. You have understood now? So is it benefit or loss? It is loss. It is loss on part of the plant by loss of carbon dioxide, by less making of glucose molecule. Definitely it is a loss. And one more thing is that it liberates carbon dioxide at the expense of ATP. I wrote it here, you see. It liberates carbon dioxide at the expense of ATP. That is, it is spending ATP, losing carbon dioxide. We know how much it will take time, energy for plant efforts to make ATP, right? So it is not only losing ATP but also carbon dioxide. So it is considered to be a waste process, right? That is the photorespiration. But according to recent research, it is revealed that photorespiration is there to defend the plant, to protect the plant. How? How does it protect the plant? You know, when light intensity increases, we have a phenomena called as chlorophyll A gets photooxidized. At higher light intensity, chlorophyll A gets oxidized. So when chlorophyll A gets oxidized, it get bleached. Photo bleaching it's called. Solarization also it is called. So chlorophyll A molecule will get disorganized and denatured. Once it denatured, you know, it is the main reaction center, it cannot perform photosynthesis. So that glucose molecule cannot be synthesized by plants. So photorespiration is a different mechanism where it tries to protect chlorophyll A from photooxidation. This is the modern review of photorespiration, right? This photorespiration takes place in three cell organelles. We know photosynthesis takes place only in chloroplast, but photorespiration takes place in chloroplast, peroxisome, and mitochondria. In mitochondria, the so-called decarboxylation takes place. What I was telling you for the last few minutes, decarboxylation takes place in mitochondria. Yes, this is the question. Now, let us have a look at the question. The oxygenation activity of Rubisco enzyme in photorespiration leads to formation of. Yes, I write once again here. One is 3-PGA. This is glycerate, phosphoglyceric acid. Another is phosphoglycolate. This is glycerate. Another is glycolate. So glycolate is 2-carbon organic acid. Glycerate is 3-carbon organic acid. Okay. So these are the products of photorespiration that are formed, right? Now, let us see the question here. He said 2 molecules of 3-carbon compound. That's wrong. 1 molecule of 3-carbon compound. Yes, 1 molecule of 3-carbon compound is there. But 1 molecule of 2-carbon compound is also there. That is not mentioned here. So let's wait for go to third option. 1 molecule of 6-carbon compound. No, it's wrong. 1 molecule of 4-carbon compound. No, it's wrong. Is also wrong. Then what is right? 1 molecule of 3-carbon compound. This is right. Actually, he has to give here 1 molecule of 3-carbon compound plus 1 molecule of 2-carbon compound. That is phosphoglycolate. This is phosphoglycolic acid. So this is the answer for this question. And one more interesting point is that this so-called photorespiration does not take place in C4 plants. Why? Why? Because C4 plants keep the Rubisco in airtight chambers. You know, Rubisco has got one weak feature. What is that? When CO2 concentration goes down, Rubisco will try to fix oxygen instead of carbon dioxide where photorespiration takes place. So what happens here? What the C4 plants did, they are very much hushar, very much clever there. What they did, they kept this Rubisco inside Bundy cells. Yes, it is quite interesting. I would like to explain that in C4 plants, The so-called photosynthesis takes place in two cells. One is mesophyll, another is bundle cell. We know mesophyll, the tissue in between outer upper and lower epidermis is called as mesophyll. Then what are bundle cells? The cells which are surrounding the vascular bundle, especially in leaf, are called as bundle cells. So, what is speciality of this bundle cells? Let us suppose this is the vascular bundle. I am drawing here, you see. This is a vascular bundle. We know that in leaf vascular bundle, upper xylem, lower phloem. This is xylem, X-Y-L-E-M. And this is phloem. Generally, xylem is always inwards, phloem is always outwards. You take any stem, dicot stem or monocot stem, xylem is upwards, phloem is downwards. Sorry, xylem is inwards, phloem is outwards. But in case of leaf, it is reverse. That is, xylem is outwards, phloem is inwards. That's one thing you have to remember. And next, around the xylem, we find the vascular bundle sheath cells. These are the bundle sheath cells. These bundle sheath cells are many in rows. They are bigger in size. They are thick-walled. They are compact, no intercellular spaces, and they have suberized cell walls. They are impermeable to the gases. They are impermeable to gases. Right? Now, in this bundle sheath cells, the so-called Rubisco is present. Do remember, they contain chloroplast also. And this chloroplast is egranal chloroplast. That is egg granule. Egg granule means chloroplast without grana. Do remember. Chloroplast without grana. We know that in grana, light reaction takes place. Grana nothing but thylakoid membranes, halo tubules, which are present one above the other like stalk of piles called as grana. In that grana, light reaction takes place where it is pigmented. So, here around the bundle sheath cells, in the bundle sheath cells, egranal chloroplasts are present. Chloroplasts without grana are present. Whereas in mesophyll, you see here in mesophyll, in this mesophyll, granule chloroplasts are present. So here, make a note, granule chloroplasts are present in the mesophyll cells and egranal chloroplasts, do remember, egranal chloroplasts are present in the bundle sheath cells. So, we find here chloroplast dimorphism, that is two types of chloroplasts are found in these plants. So what? See here, carboxylation takes place twice, primary in mesophyll cell and secondary in bundle sheath cells. You see, primarily the carbon dioxide is accepted in the form of bicarbonate ion by PEP, phosphoenol pyruvate, a 3-carbon organic acid, as it accepts the carbon dioxide and forms oxaloacetic acid. Oxaloacetic acid is formed here, that is 4-carbon organic acid. Hence, the cycle C4 cycle, and the plants are called as C4 plants. It is discovered by MD Hatch and CR Slack. MD Hatch and CR Slack. So it is called as Hatch-Slack cycle. It is called Hatch-Slack cycle. Right. So now in this oxaloacetic acid, whatever is the prime product of the primary carboxylation by PEP, that will convert into malic acid. By reduction, it will convert into malic acid. Reduction means here NADPH will convert into NADP. NADPH plus H plus will convert into NADP plus, and this malic acid will now move into bundle sheath cells by plasmodesmata. We know that plasmodesmata are cytoplasmic strands that traverse from one cell to the other through cell wall. So through the plasmodesmata, malic acid will move into the bundle sheath cells. This area is bundle sheath cells. And here malic acid undergoes oxidative decarboxylation. It undergoes oxidative decarboxylation. Oxidation means loss of hydrogen. Decarboxylation, loss of carbon dioxide. The hydrogen accepted by NADP plus to convert to NADPH plus H plus. So here NADPH is consumed. NADPH is here formed. Right? I think you are following very well. You require a lot of patience to understand all this. I am explaining C4 cycle now. In mesophyll cells, NADPH is consumed. In Bernese cells, NADPH is produced. This takes place by Malic enzyme, that is Me. Now what happens? Decarboxylation, that is loss of carbon dioxide. This carbon dioxide accepted by RUBP to take place the C3 cycle in the presence of the Rubisco. So C3 cycle in C4 plants takes place in bundle sheath cells. The exose formation takes place in C4 plants in bundle sheath cells. So here one RUBP accepting one CO2. Like that, if 6 RUBP accepts 6 CO2, then so-called one glucose molecule is formed as net product of the Kelvin cycle. So let us come to the malic acid. After losing the hydrogen and carbon dioxide, a 3-carbon organic acid, that is nothing but pyruvate, is formed here. This pyruvate will move into again the mesophyll cells, where it converts into the PEP, that is phosphoenol pyruvate, by consuming 2 ATP molecules in the presence of pyruvate dikinase. That is, for every 1 CO2, 2 ATP are extra consumed here. So total here for 6 carbon dioxide. Why do we take 6 carbon dioxide? To form one exose molecule, Kelvin cycle has to take place 6 times. So for the occurrence of 6 Kelvin cycles, 6 2s are 12 ATP molecules are consumed extra. Actually, 1 Kelvin cycle requires, you know that 1 Kelvin cycle requires 18 ATP plus 12 NADPH plus H plus. These are required for Kelvin cycle. That too 6 times Kelvin cycle takes place to form one exose molecule as the net product. But in C4 cycle, what happens? Extra 12 ATP molecules are consumed. Extra 12 ATP molecules are consumed. I write here extra. So total here 12 plus 18, 30 ATP molecules are consumed. Do remember, total 30 ATP molecules are consumed. Right? Now one more question, why 2 ATP are extra consumed in C4 cycle? That is to form the PEP. Why should it form PEP? For fixation of carbon dioxide primarily. Why should it fix carbon dioxide primarily? Because you know that Rubisco present in bundle sheath cells. So who is pumping carbon dioxide to Rubisco? PEP is pumping. PEPK is pumping. Right? If there is no PEP, for example, imagine for a while, if there is no PEP, there is no primary carboxylation. When there is no primary carboxylation, who will send carbon dioxide to bundle sheath cells? You may be surprising, sir, where it is sending carbon dioxide. It is sending carbon dioxide in the form of malic acid. Right? So who will send malic acid to bundle sheath cells? PEP is sending. If there is no PEP, no primary carboxylation. When there is no primary carboxylation, there is no oxaloacetic acid, there is no malic acid, and no pumping of malic acid into bundle sheath cells. Then when there is no malic acid, no oxidative decarboxylation of malic acid, then no CO2 available to Rubisco. You know that when Rubisco doesn't have CO2, it acts as again oxygenase. To avoid Rubisco from acting as oxygenase, there should be continuous pumping of carbon dioxide to bundle sheath cells. That is done by PEPcase from mesophyll cells. So to produce PEP, 2 ATPs should be used always to 2 ATP should be consumed always, so that it is spending 12 ATP extra for one exose molecule. So total it is 30 ATP it is sending to form one exose molecule. And moreover, they are preventing Rubisco from acting like oxygenase. So what they are preventing, in fact, they are preventing photorespiration, they are preventing loss of carbon dioxide, they are preventing loss of ATP, they are enhancing carbohydrate productivity, they are increasing yielding of the plant. By all these, what with our understanding is that C4 plants are more evolved than C3 plants in all terms: consumption of water, loss of carbon dioxide, yielding of carbon dioxide, productivity of carbohydrates. So in terms of all these, in terms of temperature also, that tropical, they can sustain high temperature C4 plants. So in terms of all these, we come to one conclusion that C4 plants are more evolved than C3 plants, and they prevent what? Photorespiration they prevent by keeping Rubisco in airtight chambers, namely bundle sheath cells. So that is what I wanted to explain you. Hope you could have understood the concept of this question, right? So you have any doubt, please drop down, please make the comment, ask me the doubts. I'm ready to explain you again all these. You want me to slow down, if you want me to get a fasten, everything I can do according to your will and wish. This channel is aimed at clarifying your doubts clearer, and get you botany nearer, right? Once you feel that yes, botany is excellent, I am understanding, I like botany, then think that 180 marks are they are present in your pocket. Okay, so do subscribe my channel Vivid Botany. Do remember Vivid Botany, and drop your comments in my channel, and let us wait for the next question. Thank you.